Blood is the largest organ in our body. 10 pints of life force coursing through thousands of miles of arteries, veins, and capillaries. Those blood vessels carry blood to every other organ, keeping them functioning and thriving.
Hi, my name is Hailey Filler.
And I'm Stephanie Maxwell. We are here to discuss the applications of chemistry in blood. We will be covering 6 main topics out of the grade 12 chemistry curriculum. These topics include organic chemistry, intermolecular forces, solubility, thermochemistry, equilibrium and Le Chatelier's principle, and buffers.
Organic chemistry. Biochemistry is the study of organic compounds and reactions that occur in living things. Biochemistry studies biological polymers, which are proteins, starches, and nucleic acids, and also studies lipids. Polymers are very long molecules that are made by linking together many smaller molecules called monomers. Polymers found in living things are called biological polymers. Biological polymers include proteins, starches, and nucleic acids. Proteins are the most diverse molecule in living organisms and accomplish more tasks than any other organic compounds in the human body. Biological polymers are composed of monomers called amino acids. In the human body, there are 20 essential amino acids that all contain a carboxyl group and an amino group. Amino acids, which are monomers, are linked by amide bonds to form proteins, which are polymers. One of the most important proteins in the human body is hemoglobin. Hemoglobin. Hemoglobin is responsible for transport of oxygen in blood, transporting oxygen from your lungs to your tissues, and carbon dioxide from your tissues to your lungs. Hemoglobin is a globular protein made up of 4 different polypeptide chains and a heme group with an iron center. Hemoglobin has 2 different conformations depending on if oxygen is present in the protein or not. Polypeptide chains. Hemoglobin consists of 2 alpha helixes and 2 beta pleated sheets. All 4 chains are types of secondary polypeptide structures, which are either coils or folds within the chain that can be caused by hydrogen and oxygen atoms near the peptide bonds between the amino acids. Alpha helix is characterized by tight coils, and beta pleated sheets are formed between the parallel stretches of polypeptides. A heme group is the prosthetic group of hemoglobin that carries oxygen to the blood. It consists of a central iron atom that bonds to oxygen and histine. This is histine, and when oxygen comes in contact with the hemoglobin, iron will bond to the oxygen. Hemoglobin conformation. There are 2 different types of conformation of hemoglobin. The first type is the T state, which is puckered because no oxygen is present and attached to the iron atom. And the second state is the R state, which is planar, and it favors oxygen binding.
Intermolecular forces. Intermolecular forces act between molecules or ions to influence the physical properties of the compounds. There are 4 types of intermolecular forces. Dipole-dipole force, which is an intermolecular attraction between opposite charges of polar molecules. Ion-dipole force, which is the attraction between an ion and a polar molecule. Dipole-induced dipole force, which is a force in which a polar molecule distorts the electron density of a nonpolar molecule to produce a temporary dipole. An instantaneous dipole-induced dipole force, or better known as London dispersion force, which is a weak attractive force that is present between all molecules due to temporary dipoles. There are many intermolecular forces in blood. Today I will be discussing the force called capillary action, which is the ability of a liquid to flow against gravity, where liquid spontaneously rises in a narrow space, porous material or non-porous material. It occurs because of the intermolecular forces between the liquid and the surrounding solid. This is important for moving blood throughout the body, and since blood is mostly water, which is a polar molecule, it experiences intermolecular forces within the arteries, veins, and capillaries. Solubility. Solubility is the ability of a substance to dissolve. In the process of dissolving, the substance which is being dissolved is called the solute, and the substance in which the solute is dissolved is called the solvent. A mixture of solute and solvent is called a solution. The dissolving of blood clots is one of the most important applications of solubility in blood. Blood clots are the cause of 80% of strokes and can also cause heart attacks. They can be caused by injury or disease. Blood clots are important to healing injured parts of the body. They can form on the site of injury in order to prevent excessive bleeding. Often, blood clots caused by injury can be dissolved naturally in the rest of the blood. Blood clots formed for reasons other than injury can be very dangerous. The first step for removing such blood clots is to determine its solubility, which can then be properly dissolved through surgery or medication. Anything that is breathed into the lungs will dissolve first in the blood and then will be transported all Anything that is breathed into the lungs will be dissolved first in the blood and will then be transported to all the other parts of the body. The blood solubility of an agent is related to its blood-gas partition coefficient. This coefficient is the amount of agent dissolved in blood to the amount of the same volume of gas in contact with that blood. The more blood-soluble the agent, this being a high partition coefficient, the slower the effect. For example, in anesthetics, the higher the partition coefficient, the slower the patient will go to sleep.
Thermochemistry. Thermochemistry is the study of energy involved in chemical reactions. Heat can be referred to as the transfer of kinetic energy and is transferred spontaneously from warmer to cooler objects. Humans are endotherms and generate internal heat to maintain body temperature. One way humans conserve heat is through countercurrent heat exchange systems. The arteries of the arms and legs run parallel to a set of deep veins. Warm blood flows down the arteries from the body core and cools as it flows towards the end of the limb, losing its heat to the parallel veins that are returning to the body's core. Once blood reaches the capillary beds at the end of the limb, arterial blood loses less heat to the environment. Overall, the transfer of kinetic energy, heat, from the warm-blooded artery to the cool-blooded vein allows the human body to maintain homeostatic control.
Equilibrium and Le Chatelier's principle. Chemical equilibrium applies to reactions that can occur in both directions. Le Chatelier's principle states that when a system in chemical equilibrium is disturbed by a change, such as concentration, pressure, or temperature, the system shifts in equilibrium composition in a way that tends to counteract the change of variable. The kidneys are an important part in keeping equilibrium from shifting. They work to maintain the constant composition of blood by helping to balance the various ions present. One of the most important reactions in the blood that kidneys help to control is the reaction between calcium ions and phosphate ions. The reaction is shown here. If this equilibrium changes, which is often caused by disease, it can cause problems in the body. The calcium phosphate helps to give bones their rigidity. If the kidneys remove too many phosphate ions, then, as stated by Le Chatelier's principle, the equilibrium will shift to the left. This will decrease the amount of calcium phosphate in the blood, reducing bone density, and can eventually lead to osteoporosis. In other cases, if the kidneys remove too many calcium ions from the blood, the equilibrium in the kidneys shifts to the right. Solid calcium can form in the kidneys, resulting in kidney stones, which are very painful. This type of equilibrium change occurs in other areas of the body as well. Two examples are gallstones in the gallbladder and gout in the joints. Another example of how kidneys maintain equilibrium in ions is the reaction of the bicarbonate ion. The reaction is shown here in the following buffer reaction. A change in this reaction can affect the pH of the blood, which, as mentioned before, must be kept constant. The kidneys can regulate high pH levels, a condition known as alkalosis. When high levels occur, the kidneys remove the bicarbonate ion from the blood. As this concentration is decreased, the reaction will shift to the right. When the equilibrium shifts to the right, more hydrogen ions are generated together with bicarbonate ions. As a result, pH decreases. Hemoglobin in red blood cells is responsible for transporting oxygen around the body. Hemoglobin is oxygenated in the lungs in the following reaction. Equilibrium will be maintained as long as there is sufficient oxygen present in the air. At significant altitudes above sea level, it is much more difficult to obtain the needed amount of oxygen. The result is, in accordance with Le Chatelier's principle, the equilibrium is shifted to the left, away from the oxygenated hemoglobin. The consequences for the reduced amount of oxygenated hemoglobin can be very severe, ranging from lightheadedness to death. The body can adapt over time to less oxygen in the air by producing more hemoglobin, which will make up for the less oxygen and shift equilibrium to the right. Maintaining equilibrium for all aspects of blood in the body is crucial for supplying the body with sufficient nutrients and minerals, which is necessary for survival.
Buffers. A buffer solution is a solution that either contains a weak acid conjugate base or a weak base conjugate acid. Buffer solutions have the ability to resist change in pH when moderate amounts of the acid or base are added. Weak acids or bases are used in buffer solutions, therefore most of their molecules do not dissociate and acids or base concentration remains high. The salts added are soluble in good electrolytes, therefore the concentration of the conjugate acid or base is high as well. Adding an acid or a base to a buffer solution has little effect because H3O or OH ions can be removed by the components of the buffer solution, resulting in a resist of large change in the pH of the solution. There are two important characteristics of buffer solutions. One, the pH of the solution, and second, the buffer capacity. The buffer capacity is the amount of acid or base that can be added to the solution before considerable changes occur in the pH of the solution. The buffer capacity is dependent on concentration. Many mechanisms in our blood and extracellular fluids allow our bodies to maintain homeostasis. The most important way our blood is kept under constant homeostatic control is by buffers dissolved in the blood. Our blood pH is around 7.35, and life-threatening problems can develop if the pH drops under 7 or rises above 7.5. The most important buffer in our blood is the equilibrium between hydrogen carbonate ions and carbonate ions. Dissolved carbon dioxide reacts with water to form hydrogen carbonate ions. Then, the hydrogen carbonate ions dissociate in water to form carbonate ions. Due to metabolic changes in the body, either hydronium ions or hydroxide ions will enter the blood. Excess hydronium ions are removed by combining with hydrocarbonate ions, and it is the reverse of the first reaction. Excess hydroxide ions are removed by reacting with hydrogen carbonate ions.